Session chair.
Good afternoon, everyone. We are about to commence Dr. E.K. Janakiammal Memorial Lecture as part of 34th Kerala Science Congress. We have Dr. M. Sanjapa, former Director, Botanical Survey of India, to deliver Dr. Janakiammal Memorial Lecture. Welcome you, sir. We are about to commence the program. I think EVP also will join the session. Dr. Hari, am I audible?
Yes, yes, audible.
Okay.
VPS joined.
Okay. So. Shall we start it?
I think we should get it started.
Okay.
Dr. Sanjapa, good afternoon.
Good afternoon, sir.
There,
Yeah.
So, Hari, we can get it started, no?
Yes, sir.
Yeah.
Yes,
So good afternoon, everyone. Let me invite Dr. P. Harinarayanan, Principal Scientist, KSST, to introduce the speaker. Over to you, Dr. Hari. Please unmute.
Hari, you are muted.
Yeah. Good afternoon and welcome to the 34th session of Kerala Science Congress, the memorial lecture on Dr. E.K. Janakiammal. Dr. G.K. Janakiammal was a renowned botanist and plant cytologist who made significant contribution to the genetics, evolution, phytogeography, and ethnobotany. Dr. Ammal was associated in the service of the Government of India in various capacities, including the heading the Central Botanical Laboratory of Allahabad and officer on special duty at the Regional Research Laboratory in Jammu and Kashmir. Dr. Ammal chose cytology, which is those formative years of science was concerned primarily with the nucleus and chromosomes. Ammal's pioneering work at the Institute of Coimbatore on the cytogenetics of sugarcane and interspecific and intergeneric hybrids involving sugarcane and related grass species and genera such as Bambusa is epochal. She was an original thinker and attributed the higher rate of plant speciation in the cold and humid northeast Himalaya as compared to the cold and dry northwest Himalayas to polyploidy. In order to promote excellent work in taxonomy and encourage students and scholars to work in this field, an award was constituted in her name by the Ministry of Environment and Forest, Government of India. Today we have with us Dr. M. Sanjapa, who was the former director of Botanical Survey of India and also the recipient of the award in Dr. Janakiammal's name. Dr. Sanjapa, after doing his BSc and MSc from Central College, Bangalore, has worked in various Karnataka government and national institutions and also some international institutions. After completing his MSc, he started his career at the Mulberry Center at Central Sericulture Research Training Institute, Mysore. And in 1979, he joined the Botanical Survey of India and worked in many capacities in scientist in charge of flora of India and being then head of Central National Herbarium, and the director of the BSI for almost 9 years from 2001 to 2010. He had been associated with many international institutions, including the Kew Garden, the Royal Botanic Gardens of Edinburgh, the British Museum of Natural History, the Lenin Society, the Indian Royal Society of Exchange, as a fellow of Indian Royal Society Exchange. He was also worked in the Royal Botanical Garden, Sydney, and also in Nepal, Sri Lanka, and Sudan. He is an author and editor of many books in botany, especially in the flora of India, and he has mentored almost 10 students for PhD. Dr. Sanchappa was also a member of several scientific societies and a member of many national and international committees, including that of DST, DBT, CSAR, Ministry of Environment and Forest, for many purposes. And he was a recipient of, as I already told you, was a recipient of Dr. E.K. Janakiammal National Award for Plant Taxonomy in 2007, and the Karnataka Rajyotsava State Award in 2017, and many others awards was granted upon him. We would like to invite Dr. Sanchappa, who is a right person to talk on Dr. Janakiammal, sir. And I also welcome all the dignitaries, Dr. K.P. Sudhi, the Executive Vice President of KCST, Sri M.C. Datta, the Mentor of Science of Government of Kerala, the directors of the various R&D institutes under KCST, and all other dignitaries and participants to this session. Thank you, sir. And I welcome you to deliver the talk on E.K. Janakiammal.
Good afternoon, all of you. It's a great pleasure to be with you all during this 34th session of the Kerala Science Congress. Am I audible?
Yes, sir.
At the outset, I thank Professor K.P. Sudhir, the president of the 34th session of the Kerala Science Congress and EVP of the Kerala State Council for Science, Technology and Education, for having invited me to deliver a talk on no less than the great Dr. E.K. Janakiammal, who was dear to me and to many in this country and all over the world. I have been fortunate to meet her and take her blessings way back in 1981 at Center for Advanced Study in Chennai, and also meet some of our students who worked in Kerala, particularly in the National Bureau of Plant Genetic Resources. I'm really fortunate that I could deliver a talk in memory of the great woman scientist and great daughter of India, who had obtained her first woman to obtain a PhD from Michigan University. To start with, I want to make a floral tribute to her, a rose named after her by the greatest couple who breed roses in Kodaikanal, Girija and Veeraraghavan, who aptly named a beautiful rose hybrid after Janakiammal. And before I start my lecture, I would like to share another slide, a very rare slide, a rare photograph. You can see here in this photograph, you'll see the giants of the Botanical Survey of India. All of you are aware that Madam Janakiammal, who prepared a roadmap for reorganization of Botanical Survey of India at the invitation of Pandit Jawaharlal Nehru, our beloved Prime Minister then. In the photograph, you see, this is one of the rare photographs that we have in the place where she was working at Allahabad and Central Botanical Laboratories, which is now shifted to Calcutta. In the photograph, you see in the center, the person with the tie is Dr. J.C. Sengupta. He was the chief botanist at that point of time in the Botanical Survey of India. And the extreme right and others, I don't want to go into the details because I don't want to cut down on my the lecture part. So the next one, for this occasion, I chose the subject Floristic Diversity of India, an overview, which gives you the essence of the flora documented so far in our country. All of you are aware, plants are one of the Earth's greatest resources. They are sources of food, medicine, with vast economic and cultural importance. They stabilize ecosystems, form habitats that sustain planet's animal and microbial life, and so also the human life. They are also threatened by climate change, environmental factors, human interactions. There are more than 400,000 species of vascular plants on Earth, with some 10% of them are yet to be discovered. These plants, both known and unknown, may hold answers to some of the world's health, social, and economic problems. Therefore, a full inventory of plant life is vital if their full potential is to be realized before many of these species and the possibilities they offer become extinct. This I'm concentrating more on the vascular plants, but the diversity of other groups of plants is much higher. But unfortunately, we do not have the kind of manpower to document the resources that we have in the other groups of plants. In 2002, United Nations Convention on Biological Diversity, that is CBD, adopted a global strategy for plant conservation, which in short called GSPC. This, where they adopted that by 2010, they will halt the loss of plant species worldwide by 2020, but it has to be revised again. And even after 100 years, the kind of manpower that we have, trained manpower that we have, and the kind of budget available for this kind of work all over the world, it may not be possible for us to halt the species loss unless we reduce the population growth. The updated GSPC includes 16 targets to be achieved by the year 2020. Target 1 of the GSPC states that an online flora of all known plants. This is the target. But this target, I would say that we have achieved to a large extent in the form of many databases that we have in the world today. But the documentation or the survey and documentation of the species in the wild is yet to get completed. Now, if you take a vast country like ours, if you lay a small quadrant of 10 meters by 10 meters, somewhere in the middle of or north of Western Ghats, this we have done practically in a place called Khas Plateau in Maharashtra, which is very similar to what we have in Kerala near Kannur. There is a plateau called Madiyampara Plateau, where the diversity of the plants is so high, but these plants are all ephemerals. That means they are very short-lived. They may live for 1 month or 2 months or 3 months, and within 6 months, they complete their life cycle. And if you lay a quadrant in these areas, the diversity of the species is enormous. In a small area, like say about 10 square kilometers, there are more than about 650 species. This is the kind of diversity that we have. And not only that, the first slide, in the next slide also you see the kind of diversity that we have. Now, we have such diversity of the floristic elements. At the same time, the kind of ecosystems that we have represented in our country is so high. Nowhere else in the world we have such complete, almost complete representation of all the ecosystems existing in the world. So we are most fortunate that to have such ecosystems existing in our country. And therefore, we are one of the 18 mega diverse countries in the world. I don't know, for some reason, of late, if you see the current check on the Google, I find that instead of 18 mega diverse countries, it is reduced to 17 mega diverse countries. At the same time, if you look at the 20 like-minded diverse countries, the organization, there are so many countries in addition to what we have seen in the mega diverse countries in the world. Now, India being the 7th largest in the world, 2nd largest in the Asia, situated north of equator, has about 329 million hectares of land, and Himalayan ranges from almost 8,586 meters from the sea level. Land frontier has about 15,200 kilometers and coastline of 7,516 kilometers. So with this vast South Asian country encompassing varied landscapes from Himalayan peaks to Indian Ocean coastline, sustains and supports immense diversity of plants. Now, based on the distribution of the various types of plants, you can see that the phytogeographic regions have been demarcated. 14 phytogeographic regions have been demarcated. Initially, it started with hooker, and subsequently it has been modified by various people. And then ultimately, Dr. R.R. Rao has modified further. Now, based on the distribution of both plants and animals, 10 biogeographic zones have been demarcated at the Wildlife Institute of India at Dehradun by Roger and Panwa. And these 10 biogeographic regions have been further divided into 26 biotic provinces. So this is completely based on both the distribution of plants and animals. Now we have, as I said in the beginning, we have varied geography, the landscapes, and the rainfall pattern also changes from one place to almost some 30 millimeters to more than 4,000 centimeters rainfall we have across different parts of India. And similarly, the temperatures also range from different parts of India. And then we have the elevation zones are also different, from almost sea level to what I call as ocean to sky expedition of the flora. If you look at a cross section of the flora in our country, it varies from coastal line to almost 6,560 feet in the Kanchenjunga peak, near the Kanchenjunga base. So now, for this very reason why Hooker, the man who is responsible for compiling or writing the flora of British India, chose to come to India to develop, to have an idea about the kind of vegetation and ecosystems and the kind of the species composition that is there across our country. So he entered the Indian Ocean from the Indian, the mainland India through the Sundarbans, and from there he ended up in the Eastern Himalayas, Sikkim Himalayas. Now, in addition to this, we also have modified the landscapes for various agricultural crops. And today we have about 15 agroclimatic zones across the India. So India has only 2.4% of the total land mass of the world. Located, this is very, very important, located at the trijunction of 3 biogeographic reams, Indo-Malayan on the eastern side, Eurasian and Afrotropical on the western and northern side. So over 13% of the recorded global floristic diversity is represented in India. One of the, as I said in the beginning, one of the 18 mega diversity countries in the world, and 3rd in Asia, 11th in the world. shares 4 of the 36 global hotspots that we have in the world. The biodiversity hotspots. When I say hotspots, means biodiversity hotspots. What are these hotspots? Hotspots are the regions or the areas that contain about 1,500 species of vascular plants, that is less than 0.5% of the global record of the species as endemics. And most unfortunate thing is it should have lost 70% of its original habitat. So Earth's biological richest areas are these hotspots and most endangered terrestrial ecoregions. So we have 35 biodiversity hotspots in the world. These hotspots, which cover about 2.3% of the Earth's land surface, contain more than 50% of the world's plant species and 42% of the all terrestrial vertebrates. That means in all put together, nearly 70% of the biota are represented in these 35 biodiversity hotspots. So these areas are which are suffering biodiversity loss and where attention is most needed. So these are the 4 hotspots which you see on the screen, which we share with the different adjoining countries in our in the Indian subcontinent. First, the Himalayas, which we share very small bit with Africa, Pakistan, Nepal, Bhutan, China, and Burma. So these are the areas where we share the Himalayan hotspot. You must be wondering as to why I am talking about this. Unless we give an idea about these various geographical features, we are able to understand why so much diversity is there in our country and why we are one of the 18 mega biodiversity countries. And the second hotspot is the Indo-Burma hotspot, that is south of Brahmaputra River, the entire northeast, Andaman group of islands of the Andaman Nicobar group, and then Southeast Asia, where we share the Indo-Burma hotspot. The most important hotspot where we are sitting today is the Indo-Sri Lankan hotspot, which we share with the Sri Lanka. And then fourth is the Sunda land hotspot, Sunda land hotspot, where only we share the Nicobar group of islands. So the range of diversity is recognized at 3 levels: the ecosystem diversity, species diversity, genetic diversity. Now, as I mentioned earlier, I will briefly deal upon the ecosystem diversity and more details on the species diversity. I am not going to speak on genetic diversity because we have not done any progress on the genetic diversity of most of our species except for the cultivated crops. Then coming to ecosystem diversity, we have forest ecosystems, then we have grassland ecosystems, then we have wetland ecosystems, then we have coastal and marine ecosystems, then we have desert ecosystems, and in deserts, we have hot deserts on the western part of India and cold deserts in the Himalayan region, particularly Jammu Kashmir, Himachal Pradesh, and also partly represented in Sikkim. Coming to forest ecosystems, the most dominant component of vegetation in India, 24.62% of the India geographical area, that is about 80.9 million hectares of India, according to Forest Survey of India 2021 report, which is significantly less than 33.3% prescribed by the National Forest Policy or International Forest Policy. Besides important ecological functions, the forest ecosystems provides food, fodder, wood, timber, raw materials for industry and non-timber products. So an invaluable resource of economically important and genetic resources. Now you look at, just to give you a feel of course, most of the Keralites have seen the best forests available in Kerala, and when they pass through across the Kerala. And these are the Shola vegetations. Again, Ponmudi, nearest place from Trivandrum, if you go to Ponmudi, you see this kind of vegetation. Then this is also Erviculam National Park, if you go, then you have this Nilkurinj flowering. And then we come down to the river systems that we have. The major rivers, although flow into Bay of Bengal across the India, Deccan Plateau, but there are several rivers, small rivers, which have been flowing into Bay of Bengal. And along these rivers, you find a riverine vegetation, which is most unique. Then we have in the Western Ghats, we have what are called as the Lateritic Plateaus. I mentioned about the Khas Plateau in Maharashtra and also Madiampara Plateau in Kannur. Of course, the plateaus are also, the low-level plateaus are there in Kasargod, in Kannur and Malappuram districts. But much lower low coastal area plateaus are also there further down, but they are not very, very important compared to what you see in the higher altitudes. So this is the kind of vegetation that we have in the month of September. If you go to Madiampara or any of the plateaus in the from North Kerala on up to southern Maharashtra, you find plateaus are brimming with such blooms, and this is one of the most interesting and most beautiful plateaus. I would say that it's a botanical paradise, I would say. Although the species diversity is less, the density of each one of those species is so enormous that it gives a sort of, it paints the entire plateau with colorful flowers. So for this, one has to wait till September, because if you go now, you will not believe that it will be a completely dried area. This is, you see, look at the balsams, that diversity that we have in these plateaus. Then coming to the moist deciduous forest, the other type of forest. I am just giving you an idea about the kind of major kind of forest. I will not go into the details because this gives you an idea as to what kind of diversity of ecosystems or forest ecosystems that we have. Then these are dry deciduous forests. Then we have the scrub forests. Then we have again the scrub forests with rocky boulders. Then we have at the coastal level the mangrove forests, which are very, very important. And the importance of mangroves have realized much only after the tsunami. Then coming to grassland ecosystems, 3.9% of that means almost 4% of the geographical area is covered by grassland ecosystems. And because of these grasslands, we have the largest livestock population in our country. Even in desert areas, the grasslands support the largest the livestock. We have semi-arid pastures of the Deccan Peninsula, which is a very large area, humid semi-waterlogged areas of terai belt of the Himalayas. Terai means the foothills of foothills of Himalayas, where the humid semi-waterlogged grasslands are there. Then we have rolling shola grasslands in the Western Ghats. You can see starting from Rajmala or Erviculam National Park, almost up to Kudremukh area, we have such beautiful shola grasslands. Then high altitude alpine pastures in the Himalayas. Once you go after 13,000 feet in the eastern Himalayas, or even much less in the western Himalayas, where you find the tree line is receding, not a single tree will grow, only the pastures will be there at the higher altitudes. And these grasslands have been divided into 5 different types based on the composition of the genera that they have, or dominant component genera that they have in the in that grassland type. So Shehima dicanthium type, Dicanthium cenchrus lasurus type, Phragmites saccharum imperator type, and Themeda arundinella type, and then temperate alpine type, which has some barley and avena. So these are some of the grasslands, originally grasslands, which are most of the blackbuck sanctuaries are in such grasslands. But over the years, there is an invasion of some of the tree species, particularly the Senegalia nilotica, which was earlier called Acacia nilotica. Then these are the shola grasslands you can see, and also the shola forest. Then we have Wetland ecosystems, of course, Kerala is well known for wetland ecosystems, like the West Bengal and the northeastern region, particularly Assam and Manipur regions, where the wetlands are very well known. And we have more than 4 million hectares of wetlands, hectares of land covered by the wetlands, both man-made as well as natural wetlands. But also there are both freshwater and saltwater wetlands. So 2.6 million hectares of man-made reservoirs, canals, fish ponds are also considered as wetlands. 2,175 natural wetlands, 65,000 man-made wetlands. And among these wetlands, there are some wetlands which have been recognized as the wetlands of international importance. So one interesting part about these wetlands is when we have to conserve these the biota or the plants, animals, and microbes associated with these wetlands, it has to be in toto. The entire wetland has to be conserved. Unless that is conserved, none of the biota will survive, only because all of them, without having the ecosystem, they will not be able to survive. We will not be able to create such a wetland outside the natural areas. So there are now recognized 46 Ramsar sites. What are these Ramsar sites? Ramsar site is a wetland site designated of international importance under the Ramsar Convention. Ramsar is a place in Iran where the first convention took place, and there the community of scientists from across the world decided to declare the wetlands of international importance, and these wetlands are named as Ramsar sites. In India, up to October 2021, we have about 46 Ramsar sites. Now, this is the map showing the Ramsar sites across the country. Then this is the Vemnad Lake or the Vemnad Coal that we have, one of the greatest wetlands in Kerala, along with the Astamudi and Sastamkota Lake. And this is another lake which I give you a few examples only. Another wetland of international importance, Kolleru Lake. It was completely demolished by the aquacultures practiced in that region. And then when the international community came heavily on then Chief Minister, and he has restored the entire lake. And today it supports almost 1 crore people. Livelihood is supported by this particular wetland. Similarly, in the northeast, we have what is called as the Loktak Lake, where these pumdis are the floating islands formed by the mat of grass species. You can see even human habitations are there in these floating islands. And again, here, lakhs of people are dependent on this particular wetland. But again, unless we conserve these wetlands with high legislations, it will not survive for a long time to come. Coming to coastal and marine ecosystems, we have realized that the mangroves are most important, particularly in the estuaries of all along the east and west coast, the Andaman Nicobar Islands, and the Minikai of the Lakshadweep Islands have the mangroves. And 14% of the geographical areas in India are covered by mangroves. 7% of the world's mangroves are in India. And the largest area of mangroves is Sundarbans in West Bengal. And Sundarbans we share with West Bengal, with Bangladesh. Three-fourths of mangroves are in Bangladesh, and only 1/4 is there with us. But it is really, if anyone have visited, it's very nicely preserved, I would say. And regeneration has been done very nicely. Replantation has been done very nicely. And thanks to Ministry of Environment and Forest, Government of India, which has funded these replantation areas of the mangroves in Sundarbans. And I wish similar programs are taken up all along the coast of east coast and west coast of India, wherever the mangroves grow. I don't say that I will not advocate growing of mangrove species wherever we want. They survive very well only in the estuaries. And we have about 14 species of sea grasses. Only recently one more species have been added. That means it comes to 16 species. These are the seagrass beds are very, very important for many of the animals. In fact, the Gulf of Mannar Biosphere Reserve, there is what is called the sea cow, which had disappeared because the seagrass beds had disappeared. Now, because of the biosphere management, they have been able to replant the seagrass beds, and the animals have migrated from Andaman back to the Gulf of Mannar. Seaweeds, there are no more than 950 species of seaweeds have been documented along the coasts of India. But still, we need to have more, intense surveys and needing ships because almost up to 14 meters depth, the algae grows, and we are yet to collect many of the algae along our coasts. Then coming to hot deserts, we have 2% of the India's landmass in Rajasthan, Gujarat, Punjab, Haryana. And also now it is extending to Delhi, almost close to Delhi, we have the hot deserts. Then we have the cold deserts, Jammu Kashmir, Himachal Pradesh, at altitudes between 4,000 and 6,000 meters. And then we have where the temperature comes down to minus 45 degree during winters, and where the rainfall is only 10 to 70 millimeters. So therefore, the vegetation, the kind of the floral composition is totally different. I said about the Champion and Shades classification of the major forest and their distribution. I'll not go into the details of this. I only want to say that there are 4 major types: the tropical, subtropical, temperate, and subalpine and alpine type of forest, which have been further divided into 16 subtypes. And I have given the distribution of that. And the tropical moist deciduous is 30.3%, and the tropical dry deciduous forest is about 38.2%. The rest of them are all limited in number compared to the size of our country, the geographical size of our country. And this is how the forest ecosystems have been classified by the Champion and Shade. And these 16 types have been further divided into 350 and odd types based on the composition of the species, like bamboo breaks and cane breaks and things like that. I'll not go into those details. So now within these forest ecosystems, we have 18 biosphere reserves, 103 national parks, 543 sanctuaries, 50 tiger reserves, 32 elephant reserves, 7 natural World Heritage Sites, 73 conservation reserves, 45 community reserves, 9 biodiversity heritage sites, 46 Ramsar sites, and 25 marine protected areas. You can imagine, in spite of this, we have not covered even we have covered just a little over 5% of the total geographical area. So for a country like ours, our protected area network should be much more than this. But at the same time, our priorities are to be checked into, because we have a large population to feed on. Now, to survey all these resources, plant resources, right from British period, that is, the Botanical Survey almost started as early as the British entry into our country, British colonies, particularly the East India Company onwards, and which established the Botanical Survey in 1890. Since then, the Botanical Survey of India, with its headquarters at Calcutta, has been surveying the country, and so far about 75% of the country has been surveyed. Still, there are many areas which have been under-surveyed, or not even set our foot on those areas at all, because they are the most inaccessible. We need modern equipment to go to such places, like even dropping by helicopter to survey some of those areas are very, very essential. And unfortunately, at a time when we have to do this, the trained manpower of taxonomy has diminished like anything at any point of time. And not only in our country, even in other countries, it has been realized. And what is called as now the CBD has taken what is called as the Global Taxonomic Initiative. That way, Kerala is the most fortunate state in the country because the best trained taxonomists are churned out from Kerala. And if you take any organization in the country, the best taxonomists are from Kerala. And we are fortunate that in an organization like the government of Kerala has taken interest in establishing so many gardens in Kerala, and the botanical research centers, institutions like JNTB JRI, then the Malabar Botanic Garden, which have been doing a yeoman service, and very good scientists are there working in these institutions. And to take name of a few of them, Dr. Satish Kumar, who himself was a greatest specialist of orchids, has done a great service to this country by working on the orchids, and who has retired a few years ago. And similarly, we have Professor Manilal, who was a Padma Shri awardee, who has churned out, he developed a school of taxonomy in the Calicut University, and the best taxonomists have come from Calicut University. Now, coming to the chloral diversity that we have in our country is, when it comes to angiosperms, that is the highest, the flowering plants, which we have done maximum work, in fact, I would say. Angiosperms are represented by 416 families in the world, and 251 of them are in our country. So out of the 3,70,000 species documented in the world, 21,849 are documented in our country. So that comes to about nearly 6% of the flowering plants of the world are in our country. Coming to gymnosperms, which is a smaller which has a smaller diversity all over the world, including our country, there are about 1,090 species documented in the world. In our country, we have about 82 species. I have put the figure 148 because the rest of them are all introduced as ornamentals in our country. So most interesting thing among these gymnosperms is about the cycads, which are considered as the dinosaurs of the plant kingdom. And these cycads, in the last one decade, we have discovered more than about 12 species, which is a very, very rare thing. But now people have, the cycads are like the elephant. So the blind people identifying elephant by its tail, by its trunk, by its legs. So this way, there is no comprehensive work done on the cycads, and now they have taken up, then we will know exactly the number of species that we have. But at present, we have about 20 species in our country. Coming to pteridophytes, the ferns and fernales, again, Kerala has taken a very good lead in this. Dr. B.K. Nair from Calicut University and his students have contributed enormously to the understanding and documentation of the pteridophytes of our country. B.K. Nair was not only in Calicut University, but also in the NBRI, the National Botanical Research Institute. And subsequently, many other people came, like Professor Beer from Punjab, have documented the number of pteridophytes. And now Botanical Survey of India, there are at least about 7 pteridologists who have been compiling all the information. Although we have brought out the checklist, but the detailed account of this group has to be worked out. So we are fairly good at the pteridophytes, gymnosperms, and angiosperms. Now comes the other lower groups of plants, the bryophytes, for example. So far there are 20,300 species documented in the world, 2,791 till, I would say, this whatever figure that I am giving here, the figures are up to December 2020. I will tell you later why it is given December 2020. Coming to bryophytes, algae, lichenized fungi, which was earlier called lichens, and fungi, bacteria and viruses. Bacteria and viruses, Botanical Survey of India is not doing any work on that, but they are only doing the compilation work of bacteria and viruses. Whereas bryophytes, algae, lichens, fungi, we have people working in Botanical Survey of India, not only Botanical Survey of India, because for a country like ours, one single organization is not sufficient. with very limited number of people, particularly in these lower groups of the plants, the bryophytes, algae, lichens, and fungi. We need the support of many of the people sitting in the universities and other botanical research institutes to contribute to this. And thanks to JNTBRI and also KFRI, we have been contributing to this aspect. And coming to algae, 44,000 species have been documented so far. 8,979 species are in India. That amounts to about nearly 17%. Again, I would say, I feel sorry that for algae, there are only 3 or 4 people in our country are left with. So the great MOP Iyengar started for the lot of work. And then in the Kerala, the Andhra University did some work in Andhra University, the coastal Andhra. And they have come out with the Central Salt and Marine Research Institute, a compiled account of the marine algae, which I mentioned in the earlier in the figure of that. But imagine algae is one group of plants which occur in marine ecosystem and freshwater ecosystem and even in terrestrial ecosystem and even grow as epiphytes on the other trees. So these are various ecosystems in which it grows, but we have only 3 or 4 people. Imagine the vast size of 329 million hectares. So this is the unfortunate part of the algae is concerned. Although Botanical Survey of India, during when I got it initiated, that at least prepare a checklist of all the plants, all the algal groups that we know. So now they have been able to complete most of the groups, the checklist. At least we know what we have. But great people like Deshikachari, MOP Iyengar, so they are the people who have immensely contributed. The Deshikachari's flora of the Sinophyceae of India has been world renowned. Then lichens are again the group which have been documented only from the places where we can easily reach. Unfortunately, again, we have a very handful of people, 6 or 7 people, and most of them are in Botanical Survey of India. We have been documenting, but there are a few who have been outside the Botanical Survey of India system who have been helping in documentation, particularly in the NBRI and also in the Agarkar Research Institute, Pune. Fungi is the largest group. So far, 75,000 species have been documented in the world, and in our country, we have 15,504 species. But again, we have very limited people. Most of the people were working on the fungi which affect the which affect the crops and other human and animal diseases, livestock diseases. So we have been concentrating more on that. Similarly, bacteria and viruses, we have been concentrating more on disease causing harmful bacteria and viruses, including the coronavirus. So this is the kind of diversity that we have in our country. That means about 5,68,077 species of all these groups are documented in the world. In our country, we have about 54,733 species documented so far. But this will go much beyond this. And we have taken almost more than 130 or 150 years to document 54,733 species. Now imagine with the kind of untrained manpower, or even if you train manpower, how much time it will take for documenting the resources of a vast country like ours. So totally we have nearly about 13% of the world's flora represented in our country. So Orchidaceae is the largest family in the world with about 28,000 species. In our country, we have about nearly 1,400 species. But again, there is a need for revision of these orchid groups, and this work is going on all over the country. Then Asteraceae, the sunflower family, is the second largest family with 24,700 species. So this is another family, because all these families are the groups of plants are economically very important groups of plants. So in our country, plants, fungi, algae, microbes, animals, all put together, we have so far documented in the last more than a century or nearly 1.5 centuries, we have documented only 205,448 species. So with the current rate, we will have to, we may take another 1.5 centuries to cover the entire country. I don't know by which time whether the ecosystems will exist, and many species have disappeared even before they are known to science. Now, this is the percentage of each group of plants, which I say we have more of people working on the higher flowering plants, and therefore the highest percentage of nearly 40% of plants that we have documented in our country are the flowering plants. The smallest number is bacteria and viruses, because for bacteria and viruses, again, very limited people. They are there only in the CCMB at Hyderabad, or Central University of Hyderabad, and NEERI of Nagpur, and very few people documenting perhaps the highest biodiversity of groups of organisms that we have in the world. So when the documentation is complete, you will see this entire table will become topsy-turvy. Bacteria and viruses will top the list perhaps, but when are we going to have such a trained manpower? I'll not go into the detail. I mean, what I want to say is almost every year 150 species, new species are being discovered. And you know where? And particularly for flowering plants, it is Kerala and Northeast India are the two places where the maximum number of new species are being discovered. Or I would say the entire Western Ghats. But again, the activity is so much in Kerala, there are large number of new species are being described in Kerala. Thanks to, again, the taxonomy schools, which the government of Kerala has supported, and they have nurtured the botanical institutions in Kerala. Now, so far, we do not have a family of plants which is endemic to our country, particularly higher plants I'm talking about. Brachycoloaceae is one family which is segregated from the Rosaceae from Sikkim, which has been described by Panigra and Dikshit way back in 2003. But again, some almost 5 years ago, when the molecular taxonomy of this particular group is done, and then it has been merged with the Rosaceae. So therefore, we do not have any species, any family that is endemic to our country. And new family records have been made from different parts of the country, particularly Northeast and Western India. Hydrotellaceae is a group of plants which is considered as a basal angiosperms in the modern APG system of classification, are found in Australia, New Zealand, and Tasmania. And after that, only in India, on the Konkan region of the Western Ghats. Now I understand it is also found somewhere in Kasargod and also in Mukambika Wildlife Sanctuary in Karnataka. These species have been recorded from various places. But the first species which was recorded is Trithuria Continents by S.R. Yadav and Janathanam from way back in 1994. Then the second family, which is not a surprise, Cistaceae, which is a desert group of plants, which is there in Pakistan, but it has been recorded for the first time from India, from Gujarat. Then Acaniaceae, Petrosaviaceae are two other families which have been recorded from Northeast India, particularly Arunachal Pradesh, because they are the adjoining Yunnan of China. These species occur, these families are distributed, and therefore they are recorded. Hanguanaceae, another family which has been recorded, which is interesting in the sense which is collected from the Vembanad coal of Kerala. It's a new family record. I presume that this could have come from the other parts of the world, particularly Indonesia, Malaysia, and there may be some, or even Sri Lanka, there may be some species which are carried by the migratory birds. Now, this is Trithuria Continents, just to give an idea as to, this looks like a mass plant, but it's very difficult to, people would have easily missed, and very keen-eyed Professor S.R. Yadav and Professor Janathanam could sit and look at these plants, dissect these plants and identify them carefully, and found it is a new species. This is a Helianthemum lippi, which is a desert species. This is Bretonidia sinensis. This is Petrosavia sakuriae, and this is Hangwana antihelmintica, which has been discovered from the Vembanad coal of Kerala. And now, most important part and the last part of my talk would be about the endemism. Endemism means the species which occur only in India and nowhere else in the world. And this is very, very important from the conventional biological diversity point of view, because we have the sovereign rights over these particular species which occur in our country. And tomorrow, if one product is developed from any of these plants, or one drug is developed from any of these plants, India will be having the final say on their proceeds, and economically, that means we can convert the biological wealth into economic wealth. This day, I am looking for this particular day, because we have documented so many species, so many genetic resources, but we have not developed even a single product which is of international standard, and which is in need, which is requested by all the international countries. And most importantly, there are more than 3,600 species of endemic to Himalayas, 250 species endemic to Andaman Nicobar Islands. There are 3 mega centers of endemism: Eastern Himalaya, Western Ghats, 25 microcenters of endemism, and I want to say that 139 plant species are known to have become extinct or only occur in cultivation. So this is the most saddest part. Though, as I said in the beginning, being a hotspot, Western Ghats has lost already 70% of its virginal vegetation. Not only Western Ghats, the Himalayas, the Andaman Nicobar Islands to a large extent, and the species, the endemic species, or even species which might have been endemic, which have not been known to science, are also disappeared from this. Now, recently, the Botanical Survey of India has brought out the endemic vascular plants of India. The other groups, we need to bring out the endemic group, endemic plants, because we don't know. Soon we may develop a product on any of these endemic plants. Now you look at this. Kerala has the second highest number of the endemic species, although Tamil Nadu has 410 endemic species. Kerala has 357 species. This number will be increased if you add up all the new species that have been recently described. So I have given the list of all the states with the number of endemic species that they have. Now, I said in the figure, when I showed the diversity of species, I mentioned about the December 2020. Every year, Botanical Survey of India and Zoological Survey of India bring out what are called as plant discoveries and animal discoveries. And these are for calendar year we bring out. And when we document all the new species described based on the material collected from India, or based on the material available in the museums and herbaria of outside the countries from India, these are, when these species are described, all of them are documented into these plant discoveries and animal discoveries. So this document is released by the Minister for Environment and Forest, Government of India, every year on the World Environment Day on 5th June. Say, you can see the last plant discoveries up to 2020 was released in June 2021, and where 187 new species have been documented by scientists from all over the country, including Botanical Survey of India. This is the reason why I said. And we have a large number of endemics. For example, this particular species, Alisicarpus belgamensis. Of course, there are many, many beautiful species in our country, but none of them have been domesticated for introduction in our gardens, in our home gardens. Nowhere you will find. Very few species have been, domestication attempt has been made. We have so many agriculture universities. We need to do this. We have given recently a project to Professor S.R. Adav in Shivaji University, Kerala, to domesticate one of the species, Delphinium mulbaricum. one of the most beautiful species of delphinium with blue flowers, which has a great demand in the, as a cut flower all over the country. But again, it has to give them kind of returns, the farmers, to cultivate them. And in some places in Maharashtra, they have started cultivating. Then look at this Cyanotis concadensis. Now, whatever I am talking about, all endemic species, which have the potential as ornamentals. Now, I am not talking about which are potential as medicinal plants and many other species. But this is, I can only speak about the ornamental species. This is Smithia hirsuta. So these are found mostly in the plateaus, lateritic plateaus across the country. Across the Western Ghats. This is Murdania laniginosa. These are the Ceropegia vincifolia, which are varied colors, beautiful, attractive flowers. But again, a lot of research has to go into this before they are domesticated. These are again Ceropegias occulta, Ceropegia jaini, all Ceropegias. There are about nearly 55 species of Ceropegias documented in our country, and 30 species, nearly 30 species are endemic to our country, and that to northern Western Ghats. So we have such vast potential diversity that we have in our country. Look at this Pinda concadensis. It's a monotypic. Of course, recently another new species have been described called Pinda syhedria. And this species is another very beautiful. This is from the natural areas I have collected. The left one is the blue flowered one, Delphinium malabaricum, which are where package of practices have been developed for this particular species for cultivation. The right side one is Pogostemon deccanensis. If you go to Kasaragod and further north, you will find this particular species growing in the paddy fields after harvesting, or in the plateaus, lateritic plateaus. One of the most beautiful looking species. This is again endemic to the Western Ghats. Thank you very much for your patient listening. Taxonomists still have a massive task to undertake: survey, identify, describe, name, and classify new species. So thank you very much for giving me this opportunity, Professor Siddiq.
So thank you, sir. We'll have just one or two questions, that's all.
Yeah, sure.
Yeah. If you have any question, just one or two, please. If anybody has a question, you can just unmute and ask the question now, or you can type in the chat box.
At least somebody can ask me a question on what these fruits are.
No questions, sir. I think then we will wind up the session, sir.
Okay.
It was an excellent talk from you, and I request, sir, one question is there, sir.
Yeah,
what
please.
would be your suggestion grooming well-trained manpower in taxonomical area of our country?
What will be the?
What would be your suggestion in grooming well-trained manpower in taxonomical area of our country?
First of all, the governments have to create more positions for these people in different parts of the country. That is the most important thing. They have to dedicate some budget for this particular purpose. Then only it is possible for this trained manpower to be absorbed, and they will be happily going into even remotest part of the country to document the resources. And it is these document the remote areas that we need to document.
Okay, since we have a short of time and we have another memorial lecture now, I request Sri Engineer Arun Kumar from NatPac to propose vote of thanks.
Okay, sir, am I audible?
Yes.
Yeah.
Oh, respected dignitaries, it is indeed my privilege to have been asked to propose vote of thanks on this occasion of 34th Kerala Science Congress for E.K. Janakiammal Memorial Lecture. We had with us Dr. M. Sanchappa, former Director, Botanical Survey of India, who is the right person to deliver a commemorative lecture on E.K. Janakiammal. He pointed out the biological importance of wetland, especially Ramsar wetlands, which are applicable to Kerala also. He has thrown light on the good practices and examples such as reclamation of Sundarbans, etc. I express my sincere gratitude to Dr. M. Sanchappa for delivering the commemorative lecture. Thank you, sir.
Thank you. Thank you.
I express my sincere gratitude to Dr. K.P. Sudhir, Executive Vice President of the KSCST and directors of all other R&D institutions for participating in this commemorative lecture. Thank you, sir. And I extend my very hearty thanks to Dr. P. Harinarayanan, Principal Scientist KSST, for welcoming and introducing the speaker for the session. Dr. Hari very well briefed us about the scientific contribution of Dr. E.K. Janakiammal also. Finally, I convey my thanks to all delegates for their active participation, which is truly helpful in successful conduct of the session. Thank you, sir.
Thank you. Now over to Bhinisha for the session.
Yeah. Thank you, Dr. Hari. Now we will move on to the next commemorative lecture. We have Dr. G.N. Ramachandran Memorial Lecture. We have with us Dr. R. Shankaranarayanan, Group Leader, Structural Biology, CCMB, Hyderabad. Now, let me invite Dr. K. Vijay Kumar, Senior Scientist, KSSTE, for introducing the speaker.
Thank you.
Over to Dr. Vijay Kumar.
Bhinisha, Dr. Sanchappa may be requested to unshare the screen.
Okay, okay.
Okay.
Yeah. Sir, please. Thank you, sir.
Thank you, Bharat.
Over to Dr. Vijay Kumar.
Stop sharing, sir. That's it. Okay. Now it's okay.
Good afternoon, sir.
Yeah, good afternoon. Respected Executive Vice President of Kerala State Science and Technology Council. Today, we are having the third memorial lecture, Dr. G.N. Ramachandran Memorial Lecture. Sundaram Narayana Iyer Ramachandran was an Indian physicist who was known for his work that led to his creation of Ramachandran plot for understanding peptide structure. Dr. Ramachandran was born in Ernakulam. After completing his BSc Honours in Physics at St. Joseph's College, Tiruchirappalli, he joined the Indian Institute of Science, Bangalore, in the Electrical Engineering Department in 1942. So he recognized his field of interest and he switched to Department of Physics in IISc to complete his master's and doctoral thesis under the supervision of the Nobel laureate Sri C.V. Raman. After completing his PG, he received his DSc in 1947. And Dr. G. Ramachandran was the first to propose a triple helical model for the structure of collagen. He also made other major contributions to biology and physics. So G.N. Ramachandran is one of the most important scientists who had given very much contribution to the science. And today, her commemorative lecture is given by Dr. Rajan Shankaranarayanan. He is also a well-known structural biologist from Center for Cellular and Molecular Biology, CCMB, Hyderabad. Dr. Rajan Shankaranarayanan is an outstanding scientist and group leader at the CCMB, Hyderabad. He has his BSc Physics from Madurai University in 1988 and MSc Physics from Madurai University again 1990 and a PhD from Molecular Biophysics Unit of Indian Institute of Science, Bangalore. After his doctoral research, he joined with Professor M. Vijayan at Molecular Physics and he conducted postdoctoral degree there. And then at the Laboratory de Biologie Structure, IGBMC, Strasbourg, France. So he conducted his postdoctoral research there at France. And Dr. Rajan Shankaran is an outstanding scientist and he won many awards. He won gold medal from Madurai Kamaraj University in physics branch. And he was selected for Shamarok Kaikini Gold Medal Award for the best thesis from Indian Institute of Science, Bangalore. In 2020, he won the Infosys Prize in Life Science category awarded by the Infosys Science Foundation. G.D. Birla Award for Scientific Research for the year 2017. Sun Pharma Research Award for 2016, Medical Science of Basic Science category. And the most important for us is that he was awarded with the Dr. G.N. Ramachandran Gold Medal for Excellence in Biological Science and Technology by CSIR India. That is very important for us because he is on the very same subject of Dr. G.N. Ramachandran. And he awarded the J.C. Bose National Fellowship from Science and Engineering for CERB of India. In 2011, he awarded Shanti Swarup Bhatnagar Prize in Biological Sciences. And also he awarded B.M. Birla Science Prize in 2018. And he won many other awards there. And there is no time for explaining all these. And he is a fellow of all the three national academies, fellow of National Academy of Science, Allahabad, fellow of Indian Academy of Sciences, Bangalore, fellow of Indian National Academy of Science, New Delhi. And he is a member of many editorial board of journals, including journal Alive, Frontiers in Molecular Bioscience, current opinion in Structural Biology, the associate editor of the Journal of Structural Biology, and an editorial advisory panel of Biochemical Journal. He has published more than 71 research papers in peer-reviewed journals. And also, he has published more than 13 reviews, news articles, and book sections. He delivered many invited talks in national and international conferences and chaired many technical sessions. So this day, we are fortunate to have Dr. Shankar Narayan with us for giving this lecture, G.N. Ramachandran Memorial Lecture. So we are proudly welcoming you, sir, to this function to deliver the memorial lecture in the name of Professor G.N. Ramachandran. Also, we are welcoming all the members who are attending offline and online in this memorial lecture. Once again, welcoming all, and please, Dr. Shankar Narayan, sir, for the memorial lecture, please.
Okay, so can you hear me well?
Yes, sir. Yes.
Oh, great. Okay, thank you, Mr. Jaykumar, for that very kind and elaborate introduction. So I also thank the organizers of the Kerala Science Congress, which I assume must have been a wonderful fair. It's unfortunate that we are not able to attend it in person this time. I hope one day I will be able to make it, in person. So with that, let me see if I can share the screen and one of you can tell me if that works. Are you able to see my slide?
Yeah, yes, sir.
Okay, very good, very good. So as I said, first of all, I am very, very delighted and also privileged to be delivering this Professor G.N. Ramachandran Memorial Lecture. First and foremost, I am actually a direct product of the department which Professor GNR started at the Indian Institute of Science way back in 1971. And this department is called, at that time it was a unit. It's a department now, which is called the Molecular Biophysics Unit. And whatever I am today in terms of my scientific achievements, the credit goes to that department and the training that I have received from that place, which was started by Professor GNR. Secondly, even today in our laboratory, we derive new insights by referring to his discovery, which is the well-known and now very famous Ramachandran map that he generated for explaining protein structures and function. And thirdly, I will be talking to you on a topic today, which is very close to Professor GNR's heart, that is on proteins. And of course, the focus of the talk would be on how proteins are made in the cell with minimal errors so that they can attain a proper shape, correct? The proteins need to attain a shape for it to function. So that is of, a subject which is very close to Professor GNR's heart. And lastly, the name GN Ramachandran, the G in that refers to a place called Gopala Samudram. The Gopala Samudram is on the banks of a small river by name Tamirabarni in the southern part of Tamil Nadu, which some of you may be familiar with this district called Tirunelveli, which is not too far away from Trivandrum. And I happen to come from another small village, which is little upstream of that. So I have more than one reason to be happy delivering this keynote lecture. So as the title suggests, what I am going to do is how proteins are made with minimal errors and how a process that we particularly looked at has helped in understanding how the eukaryotic evolution can be linked to these processes. And that's what I'll be talking to you today. So if you look at the central dogma, correct, which I'm sure all of you know, which is to say that the DNA is converted to RNA and eventually to proteins. Each of these processes, if you look at, are made with certain errors, correct? And originally, when people looked at these processes, which is just at the time of central dogma, we felt that the errors have to be minimized to the lowest possible extent. But the current understanding over the last few years particularly, what we feel is that the errors are also tightly regulated. So the biological systems try to have minimal errors, but during certain scenarios, the errors get upregulated, or you get more errors in all these processes, which allows it to evolve in a better way under certain stress conditions. So what I wanted to say is proofreading is therefore essential for any cell's survival, starting from the simplest of organisms like bacteria to all the way to humans. And it's not true for biological systems that even, in our own normal day-to-day life, and particularly you must have heard of these copy editing, which happens routinely in the context of the press, correct, which are very, very important. Otherwise, you can end up with disasters, as has been shown in this particular slide. Now, coming to protein making, so I'm sure all of you know that the proteins are made at this big protein making factory called ribosome. And the proteins are made of 20 essential amino acids, 20 amino acids. And these 20 amino acids are depicted here. And if you look at them, their sizes and shapes of most of them are quite different. Say, for example, this is the structure of what we call as an amino acid called phenylalanine. This is another amino acid called glutamic acid. So if you look at their side chains, correct, they are very different. And they operate very differently in terms of the enzymatic selections. And therefore, the enzymes that actually pick them up for putting in the protein, in the proteins, do not have that much difficulty because of their differences. However, you have some amino acids as shown here, like isoleucine and valine, or threonine and serine as shown here. They are very similar in terms of their chemical structure as well as shape. So the enzymes that pick them up and then put them on top of a carrier molecule called transfer RNA, which are eventually taken to the ribosome for protein building, make more errors. And these many errors are not acceptable, because if you make so many errors, then that is going to create a serious problem. So this fundamental problem of what we call as amino acid selection or discrimination was proposed way back by none other than the great man Linus Pauling in the 50s. And during those times, mind you, there was absolutely no information on protein structure and the link from protein structure to function. But still, he kind of proposed that if you look at some of these amino acids as shown here, the 3 pairs, they are so close in terms of their chemistry that he said that how are these molecules going to be selected during protein making, or what we call as translation of the genetic code. And as can be seen from this picture, once an amino acid is attached to this carrier molecule called transfer RNA, the molecule is carried to the ribosome. What is checked at the level of ribosome is the codon-anticodon interaction. So this is the anticodon, which are 3 bases of the tRNA. I'm sure many of you are familiar with the central dogma and the codon table. And the mRNA comes with the codon. So the codon and anticodon pairs. Nothing is checked at the level of what is the amino acid that is attached. What is checked is at the ribosomal level is majorly the codon-anticodon pairing. Therefore, if the synthetases make mistake on top of tRNA, those errors actually percolate to proteins. And in fact, this property of the ribosome is also used by many synthetic biology approaches to put unnatural amino acids on top of these molecules so that you can get desired property of an amino acid at a particular place in a polypeptide chain. So what people figured out over the next 50 years till 2000 is that these molecules require an activity called proofreading or editing. So once an amino acid is attached to a tRNA, this is the transfer RNA molecule, and tRNA synthetases play the role. So there is an enzymatic activity that puts the amino acid on top of the tRNA. This is the end of the tRNA in which the amino acid is attached. Let me see if I get my... Sorry, I was just trying to get the... Okay, so, I hope you can see the pointer here. So this is the place wherein the amino acid is attached. And if the wrong amino acid is attached, then what happens is that the CCA end or the termini of the tRNA switches to another protein module, which we call as proofreading or editing domain, and wherein only the wrong amino acid is bound, and thereby the wrong amino acid gets removed from the tRNA. So by doing this proofreading process 2 times over, that you have better accuracy during protein making. So this is what was known till 2000. And the importance of this proofreading was not very well appreciated. So a pioneering work, let me see. So a pioneering work from the group of Professor Paul Schimmel from Scripps kind of showed that if you have mutations or changes which happen in the proofreading module, which compromises the proofreading activity of these editing modules, they can result in protein misfolding. So what will happen is, when the genetic code dictates that you have to put one amino acid in a particular place, now instead of that amino acid, you will have a different amino acid because of the mistakes that are made during this process. And that will lead to unfolding of the protein. And when you have too much of misfolded protein accumulates, then you have a significant unfolded protein response leading to cell death. And eventually, if that happens in terminally differentiated cells, like neuronal cells, Purkinje cells, in this case, it resulted in ataxia. And that created problems so that the mice has, serious issues during its life cycle. So this was the first example where people showed the importance of proofreading, and if you have any compromise, which can lead to diseases. Now, the aspect that we started to look at when I started the laboratory in CCMB nearly 20 years back is the following. All the proofreading that people have looked at so far at that time was majorly how an amino acid, which is of the form L. So when I said amino acid in biology, we all assume that we have only one form of amino acid, which is called the L amino acid. I don't know why that doesn't work. Anyway, so I hope you people can see my cursor. Is it visible? My cursor is visible? Hello? Anyone? Okay.
Yes, yes.
Okay, so if my cursor is visible, I will not go further to that pointer. So the L amino acid is the one which we normally talk about. Even if I say amino acid, we all assume that we talk about L amino acids. However, over the last 15-20 years, there is a significant appreciation that the biological system, starting from bacteria to all the way to humans, do have D amino acids in the cellular pool. And these D amino acids confuse the protein synthesis machinery. However, there are multiple checkpoints in the cell that ensure that these D amino acids, which are mirror image counterparts of the L amino acid, do not enter the protein synthesis. So there are active mechanisms by which these are removed. It's not that the D amino acids are not there, as many of us had assumed several years back. So if I now look at a protein, correct, and in terms of what would happen if I make a protein with D amino acid, there is absolutely no problem. The protein would be also a mirror image of the L amino acid protein world. As has been shown here, this is the L amino acid protein world, and you can have a mirror image D amino acid protein world in principle. And very interestingly, I touched upon the Ramachandran map here, where this is the Ramachandran map. The green dots represent the L amino acid protein, whereas the red dot represents if this whole protein chain were to be in the mirror image world, which is the D amino acid protein world, then the Ramachandran plot also would become centrally inversed from the center. So you can see this would, the green dots, which are normally the red dots, which are normally prohibited in the context of Ramachandran for normal amino acids. Now all these would take that position in the Ramachandran map if the world were to be D. Now, can you prepare a protein with D amino acid, only, and will it fold? Because this is only a theoretical consideration. And this is indeed what was shown phenomenally by Kent and co-workers in the early 90s. So this is the cover of Science. So what they did was to chemically synthesize a D amino acid HIV protease. So you have the normal HIV protease, which is of L amino acids. But these guys chemically synthesize, because the protein synthesis machinery will not synthesize a full protein for you. So they chemically synthesize this D protease. And what they found was that protein folded, and the fold is kind of similar to the L amino acid protein. On top of it, what they found was it's not just the protein which is in the mirror image world, but also the protease will now chew D peptides instead of L peptides. So the chiral inversion happens not only for the protein, it also happens for the substrate. So that is what these guys showed, which kind of suggested that in principle you can make D amino acid protein world. However, what we have for several other reasons of origin of homochirality, which I will not allude to here, nature has decided to choose only L amino acid for protein making. While you can make full proteins with D amino acids, heterochiral polypeptide is not going to be folding. And that I just mentioned, the G and Ramachandran map consideration as to why you cannot have a folded protein if you have intermittent substitution of L amino acids in a D protein or vice versa. So that will not allow it to fold. And therefore misfolding will occur, and therefore D amino acids have to be excluded from the translational apparatus for making functional proteins. And that's what the cell does. We all know that all the cells which we know of, starting from bacteria to all the way to humans, they all do this job. However, it's not a very easy job, which is very trivial. there are multiple checkpoints here, which is what I am alluding to. The first and foremost checkpoint is that of what are called amino acid tRNA synthetases, which we just mentioned before, which attach the amino acid on top of tRNA. And then comes the elongation factors, which carry the amino acylated tRNA to the protein making factory, which is the ribosome. And then the ribosome also have certain specificity for L-amino acid compared to D-amino acid. Despite the fact that they all select L-amino acids more specifically, they are all porous to D-amino acids. Which what I meant by that is that they are not exclusively L-amino acid specific systems. So each one is porous for D-amino acids. However, since there are multiple checkpoints, the D-amino acid tRNAs that are made in the cell, they hang around, correct? Because they are dead-end products, because these systems are not using them for protein synthesis. Therefore, you have a serious depletion of the tRNA pool, which is a possibility. So you have a depletion of tRNA pool. Unless otherwise I recycle these deamino acylated tRNAs, I will not get fresh tRNAs for translation. Therefore, the cell dies. Of course, if I now incorporate them into the polypeptide chain, that also leads to disaster because you will have global misfolding. So you need to have a system which we call as deamino acyl tRNA DSLAs or DTDs, which as I will be referring to, which specifically looks for the deamino acid in the cellular pool and ensures that they are removed from the tRNA so that they are neither taken for protein translation nor that the tRNAs eventually become a dead-end product. Therefore, now the tRNAs can be recycled after removing the deamino acids on top. So this is how the process happens. Now, if you look at the way from a structural biologist perspective, which is what I looked at from the beginning, is that there's only one enzyme, but it takes care of all the 19 deamino acids. It's not meant for, there are not 19 systems. There's only one system. And it also takes care of all the tRNA. So despite the variation in the amino acid chemistry or the tRNA sequence, this one enzyme can handle all of them together and ensure that only the D-chirality is looked at. So that is the structural and mechanistic understanding that we wanted to know as to how this enzyme achieves this task in a significant milieu of excess L-amino acid pool, because normally you would expect that the L-amino acid pool in a cell is much, much larger than the few mistakes that are there. So that is a mechanistic challenge that this enzyme faces. So how this DTD enforces homochirality? The homochirality, what we mean is single chiral entities for making proteins. And this is what we looked at. And as I just mentioned, these DTD is responsible for recycling these deamino acid tRNAs and eventually leading to the free tRNA so that now a fresh round of translation can begin. So several years back, nearly 10 years now, that we solved using a technique called X-ray crystallography, as we are biophysicists or structural biologists. We solved the structure through a lot of interesting, mechanistic details, which I will not go into, as to how this process happens. What I will just mention here for people who may be interested is that even though the molecule is a dimeric molecule, you have one small segment, which is a gly-cis peptide connected proline, which Ramachandran must be happy to see such kind of conformation playing a very significant role in the fundamental process of chiral selection. And based on this motive, that the whole chiral selection of the system operates, and it ensures that the D amino acids are removed from tRNA and L amino acids are never touched. Now, to summarize what we did mechanistically on this problem is shown here in this slide. The L amino acid tRNA pool, which is the excess pool in the biological system, is never touched. The D amino acid pools are removed. So we wanted to know there is this amino acid called glycine, which is neither L nor D, which is an achiral amino acid. So what is happening? Because that will tell us whether this is a D selection system or a L rejection system. And we showed that this DTD has additional function of acting on glycine, which is attached to tRNA, a different tRNA, suggesting that this enzymatic module is a L rejection system. So which means anything but L can enter into the pocket, and therefore that will be removed. So this is what we showed several years back. Now I am coming to the evolutionary implications in the context of both animals as well as in the context of plants. So if you look at in all life forms, there is no life form that is available which does not have the function of D-amino acid removal, suggesting the importance of this process across the board, right? And if you look at bacteria, the enzyme that I was talking to you about just before is an enzyme called DTD1, which is present in all bacteria and in all eukaryotes. Archaea, which is another branch of life, have totally different version of DTD, which I will come back to a little later. And that DTD2 is present in addition to Archaea, it is only present in plants. So we asked two questions. What is the reason for having different variations, like a protein called ATD in only fungi and animals? That branch is called Ophistoconcha. Or in the plant kingdom, where you should have an additional DTD2 in addition to DTD1. So this is the question that we asked. And to cut a very long story short, we characterized this protein, which is present in all animals. We call that as Animalia-specific tRNA deacylase, or in short, ATD. We solved again from a crystallographic experimentation. We solved the structure of it at a very high resolution of 1.1 angstroms, at 1.6 angstroms. And we showed that now the GP motif, as you see here in the DTD, is switched from a cis peptide conformation to a transpeptide conformation. So there's a complete switch. And this switch, as you can imagine, I hope, so the switch, as you can imagine, creates an additional space, and that is responsible for removing another mistake. So here what we showed is, to again cut a long story short, when you have the genomes of higher systems compared to that of lower systems, you see the size of the genome obviously is much bigger. The number of tRNAs also goes from few tens in bacteria to from yeast. And when you go to animals, you have few hundreds to, in certain cases, few thousand tRNAs, okay, total tRNAs. Now, there is a lot of expansion of what are called tRNA isodecoders, which I will not have time to go to. So the number of different variants of tRNA expands in a phenomenal manner. So in all these 50, 60 years of this proofreading mechanistic understanding, people have been looking at a tRNA which is selected well, because the tRNA is a larger molecule, but the enzyme makes a mistake with amino acid. So what we identified is a totally new case, wherein the amino acid is picked correctly, but however, in the context of animals, since there is a huge tRNA expansion, tRNA set expansion, now the tRNAs pose a problem for these enzymes. Therefore, you pick the correct amino acid, but you pick the wrong tRNA. And therefore, this ATD is responsible for creating a solution to that problem of removing alanine, which is attached to what is called a tRNA threonine. So that is what we started looking at. So then the question is, why suddenly you have to have a protein which is solving this problem, and what is its physiological relevance? So we created CRISPR-based knockout in both HEK cells as well as in ES cells. And what we showed was, under normal scenario, correct, when you grow the cells, HEK cells or ES cells, you do not have any major problem for the cellular growth. So ATD's function was kind of not required. That was pretty surprising. However, when you subject these cells to what is called oxidative stress, then the cells started dying, suggesting that the oxidative stress can create a serious problem. And we also showed, using multiple assays, I'm just showing one here, that you now have, under oxidative stress, the ATD knockout cells have significant aggregation, suggesting that you have serious proofreading mistakes that are happening, and therefore the wrong substitution is happening, and ATD is recruited in higher systems to solve the problem. So then we started looking at, when nature recruited ATD. And what we found out was, which eventually this question took us to organisms called coinoflagellates. So what are these coinoflagellates? A person by name Nicole King from University of Berkeley is a pioneer in this area. So what she has done over a period of time is to sequence several of these organisms and studied their, the organisms' behavior. And these are, sister group of animals, and they are not multicellular on their own. However, under certain environmental conditions, they come together and exhibit multicellular behavior, even though they are not multicellular on their own. And interestingly, what they figured out was many genes responsible for multicellularity appeared at the level of these organisms, even though they are not multicellular. That means the seeds of multicellularity have already been sown even before multicellularity appeared in a branch of life. And when we looked at the problem of ATD along with the tRNA, what we found interestingly was exactly at the same place where the coinoflagellates started acquiring multicellular characteristics, because one of the major consequences of having multicellularity is significant oxidative stress. And therefore, to solve that problem, nature has recruited both ATD and this tRNA to solve this problem. So to summarize this aspect of the work, what we showed some time back is you do have significant genome expansion in animals, because of which you have serious tRNA expansion, which results in tRNA misselection error. And on top of it, since ROS, reactive oxygen species, is a signaling molecule which comes out of these several cellular processes called mitohormesis, NOx enzymes, peroxisomes, which produce them, and multicellularity also leads to significant ROS. Therefore, the function of this protein, which is being done by another synthetase, has become ineffective. Therefore, ATD has been recruited or generated as a paralog in these systems to solve this problem, is what we showed. So currently we are looking at whether why nature has to create a problem in a particular point in evolution and solve the problem then and there. So we have certain kind of indications that possibly these tRNAs are rooted for purposes away from translation, and that we are probing it. Now I am switching to the last part of the talk, which is can we take 5 minutes. So what I was mentioning about is this protein called DTD2, which has an origin from archaea, but they are present only in plants in terms of their sequence. And these DTD2 and DTD1 are totally different evolutionarily as in terms of their origin. Also, they are different in terms of their sequence and structure. So they are totally independent. So why plants recruit in addition to DTD1? Why should they have DTD2 while every other organism or every other branch of life can survive with only one DTD, is the question that we asked next. So we started looking at the literature as to what is known, and what was known was quite interesting. So, these proteins were called GEKs, correct? GEK1 in the context of Arabidopsis. And of course, at that time, it was not known to be DTD2. And what people had shown by doing what are called genome-wide knockouts on the Arabidopsis of all the genes, they found that the DTD2 knockout plants are sensitive to ethanol and acetaldehyde, and eventually they showed that this is majorly because of acetaldehyde. Now, then the question that we asked was, what is the link between the anaerobic stress metabolite? Because acetaldehyde is a product of anaerobic stress, which of course in the context of plants happens a lot in the roots as well as in pollen. So you have significant production of acetaldehyde. And what is the link between acetaldehyde and the proofreading system that we are looking at is what we asked. Then we hypothesized that maybe the acetaldehyde is modifying some part of the tRNA, which is amino acylated with the D amino acid, and the DTD is possibly modifying this effect. And to cut again a long story short, what we showed was that these modifications are nothing but ethyl adducts. And these ethyl adducts are happening on top of the D-amino acid, which is attached to the tRNA. And the modification happens at the amino group of the amino acid. And the next question that we asked is, okay, now if the modification happens, so we in vitro generated these modifications in a tube and then checked whether these enzymes will have that activity. And as shown in the figure panel here, that these enzymes very nicely remove the adducts, and no other enzyme known so far in the proofreading context had the ability to remove these adducts except DDD2, which clearly suggests that the anaerobic respiration product of acetaldehyde is modifying the amino acylated tRNA quite significantly, and therefore the plant systems have to recruit the archaeal version, wherein in archaea also you have significant amount of acetaldehyde production, which leads to the recruitment from archaea this property or this function to be part of plants. And this activity we named as NIDAT or NIDAT deacylase activity, which is essential for the plants, is what we showed. Now, again, we asked similar question as we asked in the earlier case as to when actually plants recruited DTD2, or when did they recruit this activity. And again, very interestingly, these are fantastic time for plant biologists, particularly who are looking at the evolution of processes at different branches. In addition to what I showed on the animal side, there are many processes which are required for land plants. And what people have shown is that these processes, even though they are completely elaborated when the land plants, when the plants move from water to land, however, the rudimentary processes are already set in even in branches before land plants. And there are, branches called charophytes, and these are, well-known members of the charophyte branch, which are Klebsiormedium and Chara. And these organisms actually indeed have many of the adaptations that are required to make it a successful land plant. And when we looked at DTD2, which is our protein of our interest from the acetaldehyde point of view or anaerobic stress point of view in roots, what we found was that DTDs are of course present in archaea. They are all of archaeal origin. You don't have DTDs in bacteria, and of course even cyanobacteria do not have it. Then if you look at the plant ancestors, which are the algae, you don't have them in many of these algae. However, starting from what are called subaerial charophytes, starting from chlorophytes, you do see the presence of DTD2 gene all the way to land plants. So therefore, what we proposed is that there is a recruitment of DTD2 coming from archaeal origin, which eventually help the plants to counter anaerobic stress. Now, this is the summary of the work that we published recently, which kind of puts the whole problem in a summary slide. That if you think of now, organisms moving from water to land, particularly in the context of plants, that the roots face two major issues. One is that there is low oxygen in soil, therefore there is serious anaerobic stress, which results in acetaldehyde production in roots, which is well known and characterized. And in addition, the soil has significant microbial degradation leading to D-amino acids, because the microbial cell walls are made of D-amino acids, which you may know, D-amino acid peptides. So you have abundance of D-amino acid in the microbial, in the soil. So the confluence of these two, the accumulation of D-amino acids in the soil, in addition to anaerobic stress in the soil, leads to modification of D-amino acid tRNA to a significant extent. And to solve that problem, nature or land plants or land plant ancestors recruited them nearly 800 million years ago to become successful land plants, suggesting that DTD2 and the process that it does is a key process responsible for land plant evolution, is what we proposed. So this is another study which I'm not going to go into. again emphasizing the role of these chiral proofreading systems in the evolution of eukaryotic systems. People can go and look at this paper if they're interested in. So we showed here as to how the mitochondrial translational machinery has to be optimized using DTD, without which you will have serious problem in terms of eukaryotic emergence, is what we showed recently. So to summarize, what I was just trying to indicate here is that if you have an important molecule which is there with us from day one, as the case here, because these are molecules involved in very fundamental process of chirality-based proofreading, then they are actually used in different contexts, which are very important when nature wanted to make different branches as multicellularity, land plants, or mitochondria, and so on and so forth. So the ancient biomolecules are used again and again in plethora of functions, which is known in the context of protein folds, because we know for quite some time that the genomes are enriched with certain proteins, much more protein folds more than the rest, because they are very stable folds and they are there with us for a long time. And since they are stable, nature uses them again and again. So the kind of direction, we don't have any answer to it at this point of time. What we're thinking at this point is that, is there a code? Like say, for example, if you have a reliable molecule which is with us, or a reliable member who is with us in a society for a very long time, when you have a problem, you go to that reliable. So that is what is happening. So if at all nature were to go for such kind of reliable, so-called reliable molecules, is there a code that defines where, these molecules, so that one can go to it and then use it for purposes where you have important transitions? And that is something which we will look at. And if the code exists, where is it and how is it present in these systems is something which we are looking at. So with that, I conclude. I hope I have not exceeded my time beyond the point. So I would like to thank all the members of my lab, particularly this work is of two graduate students, Mazid and Santosh. Both of them have finished their PhDs and moved on, and the work is being carried out by the rest in the lab. And I thank you all for your kind attention. And once again, I thank all organizers for inviting me to deliver this Ramachandran lecture. Thank you very much. Thank you. I hope I haven't exceeded the time. It took 35 to so minutes.
Thank you, sir. Thank you very much. Dr. Benuja?
Yes, now the floor is open for discussion. Any questions or queries from a participant side? I already requested and to type that in the chat box, but no questions so far.
Okay.
So shall we conclude, sir? Yeah, to sum up, let me invite Engineer Sabita, Scientist KSST NATPAC, to propose the formal word of thanks.
Thank you, ma'am. I hope you can hear me loud and clear.
Yes, yes.
Yes, yeah.
Okay, good evening. Respected Dr. Rajan Shankaranarayanan, other dignitaries present here, colleagues, delegates, researchers, students. It is indeed a great pleasure to all of us to hear an enlightening talk by Dr. Rajan Shankaranarayanan, outstanding scientist and group leader, CSIR CCMB, Hyderabad. And he talked about the chiral proofreading and its role in eukaryotic evolution, the proofreading essential for the proofreading and its essentiality, and the problem of amino acid discrimination, the chiral proofing of and proofreading during the translation of plants, during the evolution of plants, etc. And once again, I like to thank him for accepting the invitation and delivering today's lecture on behalf of the organizers of 34th Kerala Science Congress, KCST and NATPAC. And thank you, sir. And also, I would like to convey my grateful thanks to Dr. K. Vijay Kumar, Senior Scientist, KCST, for introducing the speaker. And above all, I thank to all of you present here for listening today's lecture and for logging in and being a patient listeners during this online session. Thank you all. And also, I would like to thank those who work behind the scenes for making this program a grand success. Thank you, sir.
Thank you.
Thank you. Thank you very much. Thank you very much.
Thank you, sir.
Thank you.
Our regards to KP Gondan sir also.
Absolutely. Absolutely. I will. Yeah.
Yes, sir.
Thank you so much.
Thank you, sir.
Can I leave the
Yes, sir.
stage? Yeah. Okay.
Okay, thank you, sir. So
Thank
we have come to the end of the second day of 34th Kerala Science Congress. Now the activities or the programs of the third day include Children's Science Congress, which will commence at 10 a.m. and also postgraduate students interactive session, which will commence at 10 a.m. Both are parallel sessions. So all the invitees are requested to join tomorrow's session at 10 a.m. Thank you. Thank you so much. Anything else, Dr. Vijay?
Okay, thank you. Thank you.
Yeah, thank you.